Hello everyone, very warm welcome to ITLS Academy. Our today's lecture is Bacterial Resistance to Antibiotics. Bacterial Resistance to Antibiotics. Let's start with introduction. Bacterial resistance to antibiotics has been recognized since the first drug was introduced for clinical use. The sulfonamides were introduced in 1935 and approximately 10 years later, 20% of clinical isolates of Neisseria gonorrhoeae had become resistant. Similar increase in sulfonamide resistance were found in Streptococci, coliforms and other bacteria. Penicillin was first used in 1941 when less than 1% of Staphylococcus aureus strains were resistant to its action. By 1947, 30% of hospital strains had acquired resistance and currently over 90% of Staphylococcus aureus isolates are resistant to penicillin. Increasing resistance to antibiotics is a consequence of selective pressure, but the actual incidence of resistance varies between different bacterial species. For example, ampicillin resistance in E. coli, presumably under similar selective pressure, and Staphylococcus aureus with penicillin has remained at a level of 30 to 40% for many years with a slow rate of increase. Staphylococcus pyogenes, another major pathogen, has remained susceptible to penicillin since its introduction, with no reports of resistance in the scientific literatures. Equally, it is well recognized that certain bacteria are unaffected by specific antibiotics. In other words, these bacteria have always been antibiotic resistant. Intrinsic and acquired resistance. First point is intrinsic resistance. This suggests that inherent properties of the bacterium are responsible for preventing antibiotic action. This type of resistance is also termed innate. There are many antibiotics active against gram-positive bacteria which have no effect on gram-negative bacteria and vice versa. This intrinsic resistance is thought to be associated with the outer shell layers such as the outer membrane, which are absent in gram-positive shells. The gram-negative cell envelope is effectively impermeable, preventing certain antibiotics from reaching their intracellular targets. Second is acquired resistance. This occurs when bacteria which was previously susceptible becomes resistant, usually but not always after exposure to the antibiotic concerned. Intrinsic resistance is always chromosomally mediated, whereas acquired resistance may occur by mutation in the chromosome or by the acquisition of genes coding for resistance from an external source, normally via a plasmid or transposon. Both intrinsic and acquired resistance are clinically important and can result in treatment failure. Although acquired resistance is more of a threat in the spread of antibiotic resistance. Genetic basis of acquired resistance. Three genetic elements are responsible for acquired resistance. These three elements are chromosome, plasmid, and transposons. And mutations can occur due to single base pair change also. We will going to study chromosomal mutations, and mutations are very crucial in the step of DNA replication process, so that they cannot transfer in next generation. So mutations can occur due to single base pair changes. Resistance to certain antibiotics can arise as a consequence of mutations to chromosomal genes because of changes in the DNA sequence. Transition involve the substitution of one prime adenine or guanine for another, and therefore one pyrimidine, that is cytosine or thymine, for another. Transversion involves a change from a pyrimidine to a purine and vice versa, that is from purine to a pyrimidine and pyrimidine to a purine. Deletions results in the loss of part of the DNA sequences. Insertions add extra base pairs to a gene. Transversion occurs when a segment of the DNA is reversed, and duplication occurs when a segment of DNA is repeated. Some of these changes also results in frame shift. More extensive changes in the DNA sequence, that is deletion, insertion, transversion, can also occur. Here, frame shift mutations occur when one or two base pairs are inserted into the DNA sequence, resulting in an altered reading frame and therefore an altered gene product. Plasmids. The bacterial chromosome contains all the genes necessary for growth and replication of cells. Many, if not most, bacteria also possess additional circular elements of DNA which are capable of replicating and transferring independently of the chromosome. These chromosomal genetic elements are known as plasmids and can code for a number of properties including antibiotic resistance. In a bacterial population, under normal circumstances, it is not necessary for all cells within that population to harbor plasmid. This has the effect of avoiding the production of unnecessary gene products unless essential for the survival of population. Assuming that a subset of the bacterial population maintains such plasmid, selective pressure following exposure to an antibiotic will ensure that plasmid containing and therefore resistant cells and their progeny will survive the antibiotic challenge. Plasmids have the ability to transfer within and between species and can therefore be acquired from other bacteria as well as consequence of cell division. This property makes plasmid acquired resistance much more than much more threatening in terms of the spread of antibiotic resistance than resistance acquired due to chromosomal mutation. Plasmids also harbor transposons which enhances their ability to transfer antibiotic resistance gene. Plasmids transfer normally occurs by conjugation or transduction in vivo. Conjugation requires cell to cell contact and involves the transfer of DNA from a donor cell to a recipient cell. Transduction is a process whereby DNA is transferred by bacteriophages and plays an important role in the transfer of antibiotic resistance in gram-positive bacteria such as Staphylococcus aureus, Staphylococcus pyogenes, and the Enterococci. A third mechanism of plasmid transfer is by transformation, which is the ability of certain microorganisms to acquire naked DNA from the environment. Transposons. Transposons are mobile genetic elements capable of transferring or transposing independently from one DNA molecule to another. The DNA molecules may be chromosome or plasmid. Transposons are normally flanked by short regions of almost identical DNA sequences known as repeats. These are termed direct repeats if they lie in the same direction relative to each other, or inverted repeats if they face in opposite direction. These repeats are thought to function as recognition sequence for enzymes involved in transposition. The central region of the transposon often codes for antibiotic resistance gene. The ability of transposons to mobilize from one DNA molecule to another has led to them being referred to as jumping genes. Transposons do not require homologous regions of DNA in order to integrate into DNA molecule, unlike the normal recombination process in bacterial cells, and are therefore a major cause of the transfer and spread of antibiotic resistance genes among different bacterial species. Furthermore, it is possible for bacteria to acquire a series of transposons coding for a different antibiotic resistances by insertion in existing plasmid or the chromosome. Conjugative transposons, which can mediate their own transfer, have been described in the last 10 years. These make the transfer and spread of resistance genes more likely since they do not depend on insertion into conjugative plasmid for their mobilization. Biochemical mechanism of resistance. Many different biochemical mechanisms of antibiotic resistance have been described, though it is worth noting that more than one mechanism may be present at any one time in a resistant microorganism. This is particularly relevant in terms of the clinical efficacy of antibiotics. The acquisition of a single resistance mechanism may render a bacterium microbiologically resistant, but therapeutically achievable levels of a drug may be sufficient to overcome such resistance. The acquisition of a second resistance mechanism may be necessary to achieve clinical resistance, that is, the amount of antibiotic necessary to overcome the resistance mechanism is greater than can be achieved therapeutically. Before considering specific mechanism of resistance for particular class of antibiotic, it is worth considering potential mechanism of resistance in bacterial cells. Gram-negative bacteria possess an outer membrane which can act as a barrier to the penetration of antibiotics. The main route of entry of hydrophilic molecule is via the porins, which form pores in the outer membrane. Qualitative or quantitative alterations in these porins can result in the decreased accumulation of antibiotics. The cytoplasmic or cell membrane of gram-positive and gram-negative bacteria can also act as an exclusive barrier. Alterations in membrane structure can reduce penetration, or the presence of proteins functioning as efflux pumps can actively remove antibiotic molecules from the cytoplasm. Bacteria may produce enzymes which inactivate antibiotics, rendering them ineffective. These may also destroy or alter the antibiotic molecule. Extracellular enzymes will be most effective in inactivating antibiotics since they will be kept away from their target sites. Gram-negative bacteria can also produce periplasmic enzymes which will act outside the cytoplasmic membrane. These mechanisms of resistance rely on reducing or preventing access of antibiotic to their target sites. But other mechanism of resistance involving the target sites themselves can be considered. So guys, thank you very much for watching this lecture.